Assalamualaikum warahmatullahi wabarakatuh and greetings to everyone. In this video presentation of Advanced Immunology, we, the Group 6, would like to present our discussion on the topic of tumor immunology.
So as for introductory, cancer can be defined as a disease which characterized by the uncontrolled cell growth and acquisition of metastatic properties. Cancer can be benign, which is a particle independent growth which tends to grow slowly and not spreading to other parts, whereas malignant cancer is fully independent tumor growth, rapidly grow, invade and destroy the normal tissue and spread throughout the body. The manifestation of cancers or carcinogenesis in most cases are associated with the activation of oncogenes and or the activation of tumor suppressor genes lead to uncontrolled cell cycle progression and the activation of apoptotic mechanism. Cancer can be classified into 4 types. Leukemia is a cancer of the bone marrow, the center of blood production. Lymphoma and myeloma are the cancers of immune system. Sarcoma is a cancer of connective tissue such as bones, muscle, cartilage, and blood vessel. And carcinoma is a cancer that starts from epithelial cells such as skin. The main cause of cancer is through genetic mutation in the individual as well as from environmental force. Apart of that, it can cause us by external element which known as carcinogens. These carcinogens are physical, through radiation and ultraviolet light, chemical, which from various hazardous materials such as tobacco, smoke, alcohol and arsenic, and biological like viruses, bacteria and parasites.
Tumor immunology refers to the relationship between immune functions and tumor cells, which is crucial for our understanding of the mechanisms of both tumor rejection and tumor progression. The immunological mechanisms involved in cancer growth are highly complex, including tissue-resident and blood-derived cells. The human immune system mounts natural endogenous response to highly immunogenic tumor cells through a series of steps, including the presenting of tumor antigen T cell via antigen-presenting cells, priming and activation of T cells in the lymph nodes, trafficking and infiltration of T cells into tumor banks, recognition of cancer cells by T cells, development of antigen-specific effector and memory T cells, and humoral immunity, allowing effector T cells and other endogenous immune cells, as well as tumor-effective antibodies to tumor that eliminate cancer cells.
First, types of tumor antigen. Tumor antigen consists of 2 types, which is tumor-specific antigen and tumor-associated antigen. Tumor-specific antigen is a neoantigen resulting from somatic mutation and is expressed only in tumor cells but not in normal cell. Normal cell do not express tumor-specific antigen, thus they are considered as a non-self by the immune system, and neoantigen-specific immune response are not affected by tolerance. Targeting of tumor-specific antigen does not easily induce autoimmunity, so neoantigen are ideal targets for therapeutic cancer vaccine and T-cell-based cancer immunotherapy. Next, tumor-associated antigen. Tumor-associated antigen is a protein expressed by unmutated genes and appears to be significantly overexpressed in tumor cells but rarely in normal cells. Tumor-associated antigen are normal host protein. They are subject to both central and peripheral tolerance mechanism. Targeting of tumor-associated antigen may lead to autoimmune toxicity. Oncofetal antigen protein, they are expressed at high level of cancer cell and in normal early fetal. 2 major of oncofetal antigen, which are alpha-fetoprotein and carcinoembryonic antigen. Firstly, alpha-fetoprotein. Alpha-fetoprotein is a plasma protein that produced by the embryonic yolk sac and the fetal liver. The normal range of alpha-fetoprotein concentration in human is 0 to 20 nanogram per milliliter. This level rises in patients with hepatocellular carcinoma and non-seminal testicular carcinoma. Secondly, carcinoembryonic antigen. Carcinoembryonic antigen is a protein normally found in very low level in blood of adults. Carcinoembryonic antigen level in normal people range up to 2.5 nanogram per milliliter. The carcinoembryonic antigen blood level are often increased in colorectal cancer and may be increased in other cancer including breast cancer, pancreatic cancer and stomach cancer. Other than that, tumor-specific neoantigen. Neoantigen are mainly tumor-specific antigen generated by mutation in tumor cells which are only expressed in tumor cells. It serves as targets of spontaneously arising adaptive immunity to cancer and thereby determine the ultimate phase of developing tumor. It also functions as optimal targets of cancer immunotherapy against established tumor.
For the second point, immune mechanism of tumor rejection. Both in an adaptive immune response to tumors can be detected in patient and experimental animals, and various immune mechanisms can kill tumor cells in vitro. In this section, we will review the evidence of anti-tumor immunity mediated by T cells and other immune effector mechanisms. The principal mechanism of immune protection against tumors is killing of tumor cells by CD8 plus CTLs. The ability of CTLs to provide effective anti-tumor immunity in vivo is clearly seen in animal experiments using carcinogen-induced and DNA virus-induced tumors. CD8 plus T cell response specific for tumor antigens may require cross-presentation of the tumor antigen by dendritic cells. Disease. Most tumor cells are not derived from antigen-presenting cell APC and therefore are not present in secondary lymph organs where they can display antigen to naive T cell or the tumor cell express co-stimulatory molecules needed for naive T cell activation. CD4 plus helper T cell contribute to anti-tumor immune response by several mechanisms. CD4 plus T cell respond to tumor antigens and commonly found in animal models and patients with cancer, and the presence of T helper cells with CTLs in human tumors correlate with good prognosis. Thus, To initiate anti-tumor CD8 plus T cell responses, tumor antigen have to be presented by dendritic cells. The best antigen presenting cells for transporting tumor antigen to secondary lymphoid organs and for activating NAF T cells. Dendritic cells at the site of tumor can ingest tumor cell or their protein antigens, carry the tumor antigen to lymph node, and colocalize with NAF CD8 T cells. Furthermore, the dendritic cells can deliver the ingested proteins from phagosome into the cytosol so that they are processed by proteasomes into peptides that are then display bound to +1 MSC molecules for recognition by CD8 plus T cells. Next, macrophages. Macrophages are capable of both inhibiting and promoting the growth and spread of cancer, depending on their activation state. Classically, activated M1 macrophages can kill many types of tumor cells. How macrophages are activated by tumors is not known. A possible mechanism is recognition of damage-associated molecular patterns from dying tumor cells by macrophages in an immune receptor. Macrophages in tumor also may be activated to kill tumor cells by interferon gamma produced by tumor-specific T helper cells, CTLs, and NK cells. This may be why a large number of T helper cells in some tumors is correlated with a good prognosis. And when macrophages can kill tumor cells by mechanisms that they also use to kill infection organisms, including the liberation of lysosome enzymes, nucleosides, and reactive oxygen species.
NK cells are innate immune cells that display potent cytolytic activity in response to infected or transformed cells. Upon activation, NK cells mediate the tumor killing mainly by releasing cytotoxic perforin and granzyme by degranulation, and also triggering apoptotic pathways in tumor cells through the production of cytokines such as tumor necrosis factor alpha, chemokines, granulocyte, macrophage, colony stimulating factor, or via direct cell-to-cell contact, for instance through the activation of the tumor necrosis factor related apoptosis inducing ligand or TRAIL and fast ligand pathways. The next immune mechanism of tumor rejection is antibody-dependent cell-mediated cytotoxicity or ADCC. ADCC refers to the killing of a target cell which is coated with antibodies by an effector cell of the immune system. ADCC can be activated by several channels such as via specific binding of antibodies which involve IgG and IgE, and also by the recruitment of immune cells by the Fc fragment of the antibody which involve macrophages, NK cells, neutrophils, and eosinophils. First, the B cell produce antibodies directed against specific antigens on a pathogen or a foreign body. These antibodies bind to the target antigen, and the constant region of the antibody is recognized by the effector cells of the immune system via Fc receptors on their surfaces. Following recognition, the effector cells release chemicals which then lyse and kill the target cell bound to the antibody. There are three modes of action by ADCC. The first one is by activation of calcium-dependent granule exocytosis that can lead to the release of intracellular granules from an effector cell which target the pathogen. In exocytosis, perforin and granzyme are released to attach and form pores at the cell membrane, which then leads to the cell lysis. Second is the engagement of Fas, which activates a cascade of signaling and leads to cell death mediated by NK cells. The third one is through the release of reactive oxygen species by certain effector cells when the Fc receptors are cross-linked to induce cell death.
Literally, despite of the discovery made on the development of a specific immune response along the process of carcinogenesis, the spontaneous removal of the tumor by immune mechanisms rarely occurs. Furthermore, immunotherapy approaches strengthening natural tumor-specific immune response are also found to be clinically limited and challenging. The reason behind of these challenges are due to the presence of various strategies for tumor cells to escape from immune surveillance. At a glance, four main strategies of tumor escape mechanisms are discussed, which are the avoidance of recognition by the host immune cells, apoptosis of immune cells, immunoregulatory effect of tumor cells on leukocyte functions, and also expansions of Treg cells. First, the avoidance of recognition by host immune cells are particularly manifested by low immunogenicity and or low antigenicity of tumor cells. In this strategy, several notable molecules whose expression may be downregulated in tumor cells are MHC complex, EAA, and costimulatory molecules. Primarily, the ability of tumor cells to be avoided or out from immune surveillance is characterized with no or low expression of MHC, particularly MHC class 1, thus making them difficult to be recognized, and the initiation of apoptosis, which involves cytotoxic T cell lymphocytes, can be prevented. Even though tumor cells exhibiting low or no expression of MHC class 1 are susceptible to the action of the NK cells, they are not destroyed because NK cells have no immunological memory. Next, tumor-associated antigens or TAA is primarily expressed on the cell surface as part of major histocompetitive complex. It's in elevated levels on tumor cells, but also expressed at lower level on healthy cells. Nevertheless, for this scenario, tumor cells often lack of this antigen, which makes unrecognized by cytotoxic T cell lymphocytes in producing immune response. Besides, costimulatory molecules are the heterogeneous group of cell surface molecules that act to amplify or counteract the initial activating signals provided to T cells from T cell receptor following its interaction with an antigen or an MHC, thereby influencing T cell differentiation and fate. However, costimulatory molecules such as CD80 or CD68 are often absent from the surface of tumor cells, which makes activation of T cell lymphocytes become impossible. Another escape mechanism of tumor cells is through the apoptosis of immunocompetent cells. In fact, some tumor cells are capable of inducing apoptosis in immune effector cells. For instance, melanoma cells can release exosomes containing Fas ligand or Fas-L, which interacts with Fas molecule on the space of the lymphocyte, leading to the death by apoptosis. This process is either direct or indirect through dendritic cell binding exosomes secreted by the tumor cells. For information, exosomes are small particles of 80 to 200 nanometer in diameter, whose potential role is to transmit signal among antigen-presenting cells, while Fas molecules belongs to the family of the receptors, which are involved in initiating a cascade of apoptotic signals to these cells. Furthermore, some studies have found that Fas-L also can be released by activated NK cells themselves, resulting to their own apoptosis. Therefore, in each time an NK cell is killing a tumor cell, then it also activates its own apoptosis process and dies. Apart of Fas-L activation, apoptosis of immunocompetent cells also can be induced by immunosuppressive cytokines located in the tumor microenvironment, such as TGF-beta and interleukin-10, by changes of redox potential or by changes in the intracellular calcium or ceramide level.
Okay, the third tumor escape mechanism is by immunoregulatory effect of tumor cells on mucosal functions. As shown in this figure, tumor cells secrete a wide range of substances known as tumor-derived soluble factors, which inhibit or decrease the local immune response. These include cytokines such as interleukin-6, interleukin-10, tumor growth factor, and vascular endothelial growth factor. Their activity gradually spreads from tumor environment to adjacent lymphatic organs and peripheral blood vessels. This causes T-cell tolerance. The tolerance causes T-cells cannot differentiate between self and non-self antigens, which contributes to uncontrolled tumor growth. Next, tumors often have a potential of shifting the balance from T helper 1 to T helper 2 cells. The main role of Th1 cells is to stimulate cell-mediated immunity, while Th2 cells primarily assist in stimulating these cells to make antibodies. Th1 type of immune response is characterized by cytokines which are produced by Th1 CD4 plus lymphocytes, the main being interferon gamma and tumor necrosis factor beta, which are the chief pro-inflammatory factors. By contrast, Th2 cytokines such as interleukin-4, interleukin-5, interleukin-10 tend to produce anti-inflammatory responses. As shown in the right figure, Th1 induces immunostimulation of the leukocyte, which results in tumor regression. On the other hand, shifting of Th1 cells to Th2 cells causes downregulation of the adaptive immunity, which enhances the tumor progression. The fourth tumor escape mechanism is immunosuppressive properties of T regulatory lymphocytes and their influence on tumor development. As we know, in normal conditions, T regulatory cells function to suppress immune response, thereby maintaining homeostasis and self-tolerance. However, in tumor environment, T regulatory cells percentage tend to increase, which causes inhibition of anti-tumor immune response. The center figure shows the role of T regulatory cells in tumor. Let us see one by one. First is T regulatory cell accumulation in the tumor. T regulatory cells are recruited to tumors from the periphery or by tumor-derived or hypoxia-induced CC chemokine ligand 28, but also DC dendritic cell and tumor-derived CC chemokine ligand 22. Secondly, within the tumor microenvironment, T regulatory cells can be expanded by tumor growth factor beta and possibly interleukin-10 that can also convert CD4 plus naive precursors into induced T regulatory cells. Thirdly, T regulatory lymphocytes arising in the periphery actively suppress anti-tumor effector activity of CD4 plus and CD4 plus lymphocytes by producing specific cytokines. Fourthly, T regulatory cells stimulate tumor angiogenesis directly by producing vascular endothelial growth factor.
Section 4 discusses the immunological approaches for cancer therapy. Immunotherapy manipulates the immune system using a variety of methods and drugs. Immunotherapy can be active or passive in nature. Both of these approaches can either be specific or non-specific. First, active immunotherapy involves stimulating the patient's immune system to fight the cancer cells. Generally, there are three methods in active immunotherapy. The first method is cytokines manipulation. The agents used for these methods are interferons, interleukin, and tumor necrosis factor alpha. This method is considered as non-specific as it targets general immune response to fight the tumors. The expression of co-stimulators and cytokines will boost cell-mediated immunity to the tumors. The release of cytokines will stimulate proliferation and differentiation of a wide range of immune cells such as macrophages, T lymphocytes, and NK cells. This will help to strengthen the overall immune system to fight against the tumor. For example, interleukin-2 has been demonstrated to be capable of banishing metastatic renal cell carcinoma and melanoma. Okay, the second method is tumor vaccines. Vaccination is categorized as specific active immunotherapy as it produced from killed tumor cells or with tumor antigens or peptides. However, not every cancer has its own vaccine since it is more complicated due to the cancer cell closely resembling the normal cell, and also each person's tumor has its own unique antigen. Several cancers that are caused by viral infection have a preventive vaccine to lower the risk. For example, liver cancer that is caused by hepatitis B virus. Lastly, the third method is gene therapy. Gene therapy apparently succeeds in shrinking and even eradicating large metastatic tumors. This is done by inserting genes into a patient's own cells, which then enable the body to fight the tumors without medication. The introduction of new genes into a cancerous cell or the surrounding tissue causes the cancerous cell to die or slow the growth of the cancer. Okay, next is passive immunotherapy. This therapy involves the administration of immune molecules to patients who do not produce the immune molecule on their own. The first method is monoclonal antibodies. It is a specific drug treatment that can be designed to target certain cancer cells. This drug recognizes and finds specific protein on the cancer cells. An example of monoclonal antibody is rituximab, which is used for treatment of non-Hodgkin's lymphoma. This drug targets antigen CD20 on B lymphocytes and then induces an adaptive anti-tumor immune response. The second method is an adaptive cellular therapy, or known as T-cell transfer therapy. The cultured T-cell with anti-tumor reactivity are transferred into the cancer patient. For tumor-infiltrating lymphocyte therapy, lymphocytes that recognize the tumor cell the best are taken from the patient and then cultured in a large number in the lab, and then the lymphocytes are given back to the patient so they can fight the tumor cells. For CAR T-cell therapy, the T-cells are taken and then changed to make a type of protein known as chimeric antigen receptor that enables the T-cell to attack and kill the tumor cells. As a conclusion, cancer's malignant phenotype is caused by the error in proliferation control, tumor cell resistance to apoptosis, and tumor cells' capacity to penetrate the host tissue and spread to distant places. Basically, there are two types of tumor antigen, which are tumor-specific antigen and tumor-associated antigen. Cytotoxic T-cells, NK cells, and macrophages that were triggered by the tumor antigen-specific helper T-cells will generate the immune responses that are capable to destroy tumor cells in the immunological mechanism of tumor rejection. Oncologists and immunologists have been working on immunological approaches to treat the cancer patient for many years, but it is only recently that exciting and broadly applicable breakthroughs such as cytokines manipulation, tumor vaccines, gene therapy, monoclonal antibodies, and T-cell transfer therapy have been successfully used to treat the cancer patient.
These are the list of references from various sources which had been utilized in our video presentation. We would like to express our appreciation and gratitude for your full attention on our video presentation. With that, thank you.